Hello everyone, my name is Amber Chatten. I'm a PhD student at the University of St Andrews, looking at the effects of climate change on the fertility of marine and freshwater organisms. Today I'll be presenting the preliminary results from my project on understanding the effects of changing pH on the fertility of aquatic animals using a meta-analytical approach. Water pH is a critical factor in determining the health and stability of aquatic ecosystems, as well as overall environmental quality. Changes in pH have emerged as a significant stressor for aquatic animals, particularly in the context of the rapidly changing climatic conditions. Anthropogenic CO2 emitted to the atmosphere is absorbed by the oceans, causing a progressive increase in inorganic carbon concentrations and resulting in decreased water pH and calcium carbonate saturation. This phenomenon is called ocean acidification and is in addition to the warming effects created by CO2 emissions. pH changes can also be caused by chemical pollution from anthropogenic activities like agricultural and industrial operations. The altered pH can affect the biological and physiological functions of aquatic organisms, including respiration and oxygen consumption, protein and enzyme levels, and growth and reproduction. Uncompensated changes in extracellular pH and intracellular pH may affect the biological and physiological processes of an organism. Compensation occurs by pH and ion regulation across the epithelia of gills, guts, and kidneys, driven by energy-consuming ion pumps. Owing to the associated energetic costs, exposure to ocean acidification may cause shifts in the energy budget of cells, tissues, or the whole organism. In response to the higher energy expenditure, organisms are required to modify their physiological needs by either increasing the feed intake to suffice new high energy demand or limiting energy for crucial metabolic needs. This could lead to decreased energy for functions such as gamete production, mating behavior, and reproduction. pH changes can also impact motility, fertilizing ability, and the velocity of sperm, as well as the duration of the motility period. Changes in pH can also affect the gene expression and proportion of abnormalities in fertilized eggs in some species. At increased pH, some species also show low hatching survival rates. pH changes may affect several traits at the same time, for example, disrupting the gamete production and mating behaviors, or the effect of pH change on one single trait may have cascading impacts on other traits. Gonad development being negatively affected has knock-on effects on gamete production and quality, as losses in fertility can have a significant effect on a population's dynamic stability and ultimately its survival. It is important to consider the reproductive traits when determining the effect of anthropogenic climate change on different organisms. So, what characteristics make aquatic animals particularly vulnerable to any negative effects of changes in pH? Aquatic animals with calcium carbonate exoskeletons like mollusks and crustaceans are highly vulnerable to acidic waters. Increased carbon dioxide dissolves in seawater, forming carbonic acid, which lowers the pH and reduces the availability of carbonate ions needed for shell formation. This makes it harder for these organisms to build and maintain their shells, leading to weaker structures, reduced survival, and reproductive challenges. Ocean acidification threatens these species and disrupts marine ecosystems reliant on them. Sessile organisms are fixed in place and cannot move to areas with more favourable pH conditions. They're directly exposed to local changes in water chemistry, making them particularly susceptible to acidification. Motile organisms such as fish and some crustaceans can move to areas with less acidic water, giving them some level of protection. However, this mobility may be limited by other factors such as habitat availability or competition. Early life stages of aquatic animals, such as eggs and larvae, are particularly vulnerable to pH changes. Acidification can affect the development of embryos, reduce hatching success, and impair the survival of larvae. These early life stages lack specialized mechanisms for acid-base regulation, making them more prone to physiological stress and mortality under pH stress. Many aquatic animals, such as fish and coral, rely on external fertilization, where eggs and sperm are released into the water. Acidic conditions can interfere with this process by reducing the viability of gametes and altering sperm motility or impairing fertilization success. Tropical species are predicted to be more susceptible to pH fluctuations than subtropical or temperate species. This is because of the tropical species live in an environment where the temperature is close to their upper tolerance limit range, and any additional stresses will pose a severe impact to their life. And polar ecosystems are particularly vulnerable because cold water absorbs more carbon dioxide, leading to faster acidification. The unique adaptations of polar organisms to their environment often leave them less resilient to rapid pH changes. Each of these factors interacts and contributes to the overall vulnerability of an aquatic species to ocean acidification and other pH-related shifts in their environment. Meta-analyses are useful tools to help us understand the effects of climate change on populations, as it allows us to synthesize the current data and reveal general patterns on how fertility traits are impacted by the changes in pH. The variety of responses within and between taxa suggests that ocean acidification is a driver for substantial change in aquatic ecosystems, potentially leading to long-term shifts in species compositions. Therefore, it is important to produce a set of predictor traits for the effects of pH changes on the aquatic animal fertility. Our meta-analysis identified 212 papers on the effect of pH on fertility, using a large database of studies compiled by over 40 researchers from the European Thermal Fertility Network. These researchers identified and screened the titles of approximately 25,000 studies before fully screening and compiling a database which contains 2,333 studies. They have the data on the effect of temperature, humidity, pH, and salinity on terrestrial and aquatic organism fertility. This meta-analysis incorporates data from 66 studies and yielded a total of 582 effect sizes, distributed across various species, environmental conditions, and biological traits. The majority of the effect sizes pertain to decreasing pH or acidifying changes, with 483 effect sizes, with a smaller subset focusing on alkalizing changes or an increase in pH, 99 effect sizes. Of the total effect sizes, 362 are from marine environments, and while the 220 are from freshwater ecosystems. We extracted data on a range of fertility traits, from gamete traits such as gamete size, quantity, and quality, gonad traits like testes length and ovary diameter, and reproductive outputs including clutch size, hatchling number, and fertilization success. The data encompasses 53 species, represents 7 phyla, demonstrating substantial taxonomic diversity. Notably, chordates contribute nearly half, 49% of the total effect sizes, and echinoderms are the second most abundant group, contributing 20% of the effect sizes. This dataset provides a robust basis for evaluating the effects of environmental pH changes across a wide range of species, habitats, and traits, offering insights into the potential vulnerability of aquatic fertility to changing pH. So our hypothesis was that altered pH levels in aquatic environments would have negative effects or reduce fertility in many species, with larger deviations from optimal pH levels predicted to cause greater declines in fertility. Species that release gametes directly into the water column are likely to be more negatively affected, as their gametes are directly exposed to the external environment, making them particularly vulnerable to environmental stresses like pH change. Gametes and larvae are likely to be highly sensitive to pH fluctuations, as these early life stages also lack the specialized mechanisms for acid-base regulation, making them more prone to physiological stress and mortality under changing pH. We also predicted that male reproductive traits would be especially affected because traits such as sperm motility, which is essential for successful fertilization, depend on precise intracellular and extracellular pH conditions in many species. As increased carbon dioxide and more acidic conditions decrease the availability of carbonate ions in the water, corals, echinoderms, and mollusks are likely to be more negatively affected, as they rely heavily on calcium carbonate for the construction of their shells and exoskeletons. Tropical species are predicted to be more susceptible to pH fluctuations than subtropical or temperate species. This is because tropical species are living in an environment at their upper temperature tolerance. Therefore, any additional stresses could have a greater impact. And marine species may exhibit greater sensitivity to pH changes because their habitats have historically maintained more stable pH conditions, leaving them with limited adaptive mechanisms. In contrast, freshwater species often experience naturally fluctuating pH and may have higher tolerance. However, this may be mitigated by the insular nature of many freshwater habitats, which hampers the compensatory movements to more favorable conditions. So we analysed effect sizes that looked at acidification from a species optimum pH and those that looked at an increase of pH or alkaline change separately. So I'm going to first go through the results of the effect of acidification on the fertility of aquatic animals. This is the overall model for the effects of acidifying changes in pH. The x-axis shows the magnitude of the effect size for each point, and the size of each point shows a measure of the standard error. A negative effect size means that the fertility trait is being affected negatively by acidification, so gamete number is reduced, fertilization success is reduced, whereas a positive effect size means that the fertility traits are positively affected by acidification, so increases in gamete number and hatchling success, for example. We found no significant overall effect, with large variations in the response to acidification. So I then looked at different factors or moderators which may explain these differences in the effect of pH on fertility. As we expected larger deviations from optimum pH levels to cause greater declines in fertility, I looked at the effect of the magnitude of the pH change. Each change of 1 pH unit represents a tenfold change in hydrogen ion concentration, so we use the inverse log of the magnitude of change, so it represents the true change in hydrogen ion concentration that the organisms experience, and found that there was no significant difference in the effect of pH across different magnitudes of change. I then looked at whether habitat type may impact the effect of acidification. We found that marine and freshwater fertility were not affected differently by acidification. We had expected to find that external fertilizers would be more negatively affected. However, we found no significant difference between the effects of acidification on internal and external fertilizers. Although gametes, and particularly sperm, is thought to be more vulnerable, however, when we looked at whether the different trait categories were affected differently, we found that all traits were affected the same. We then separated the traits into further subcategories, but still found no significant difference between these subcategories. We expected gametes and early development exposure would have a more negative effect than adult exposure due to the lack of specialized mechanisms for acid-base regulation. However, we did not find that they were more negatively affected than adults. We then looked at whether male, female, or combined fertility traits were more negatively affected and found no significant difference, even though current literature suggests male traits may be more vulnerable. We expected corals and echinoderms and mollusks to be more negatively affected, and although mollusks and echinoderms had the most negative mean effect size estimates, we did not find a significant difference between phyla. We then looked at whether tropical or polar species were more vulnerable than any other species and found no significant difference across climate zones. I then looked at the pH manipulation method. I categorized this into two types, either carbon dioxide aeration or to decrease the pH, or adding chemicals like hydrogen chloride or sulfuric acid. There was no significant difference between the two acidification methods. We then looked at the overall effect of alkalization on the fertility of aquatic animals using the small subset of effect sizes we extracted. This is the overall model for the effect of increasing pH or becoming more alkaline. We found a significant positive effect of alkalizing pH changes, suggesting that fertility is likely to increase if there is an increase in pH. I then looked at the magnitude of the alkalizing change and whether that impacted the effect on fertility, and found a significant relationship. As the magnitudes get larger, the effects become more negative. As we thought this may be driven by the 4 very large magnitudes of change, we conducted a sensitivity analysis. The sensitivity analysis found that when those 4 effect sizes were excluded, there was no significant difference across the magnitude gradient. The take-home messages from this study is that there is no overall effect of water acidification on aquatic animal fertility. Also, surprisingly, we found that external fertilizers are not more vulnerable than internal fertilizers to acidification. The gametes and larval stages are not more vulnerable than adults, although we did have a limited sample size for early developmental exposure. There is no difference between the effects of male and female traits, and coral, echinoderma, and mollusk fertility was not more vulnerable than any other phylum, and there was no difference in the effect of acidification on different climate zones. Thank you for listening, and thank you for everyone who's helped with this project.